DNA replication is a biological process of producing two identical replicas of DNA from one original DNA molecule. DNA replication occurs in all living organisms, acting as the basis for inheritance. Today, we will focus on three topics. First topic is DNA replication in prokaryotes. Second topic is DNA replication in eukaryotes. Third one is DNA damage and DNA repair. Before DNA replication mechanism was known, there were three postulated methods of DNA replication. The first model is conservative model. In this model, DNA replication results in one molecule that consists of two original DNA strands and another molecule that consists of two new strands. The second model is dispersive model. In this model, DNA replication results in two DNA molecules that are a mixture or hybrids of parental and daughter DNA. In this model, each individual strand is a patchwork of original and new DNA. The third model is semi-conservative model. In this model, DNA replication results in two copies of original DNA molecules, each of which contains one of the original strand and one newly synthesized strand. Evidence supports semi-conservative model. Here, two strands in red are parental DNA strands. Two strands in gray are newly synthesized DNA strands. Following semi-conservative model, after DNA replication, two copies of DNA molecules are made, each containing one of the original strand and one newly synthesized strand. Main enzyme involved in DNA replication is DNA polymerase. Here is one of the original strands that serves as a template, and this is a newly synthesized strand. New nucleotide is added at the 3' end of the newly synthesized strand. And the result is two DNA molecules, as seen in this picture, each containing one original strand and one newly synthesized strand. Both strands run anti-parallel. Here is a template strand, 5' on the bottom and 3' on the top. And here is a newly synthesized strand. New nucleotide is added to the 3' end of the new strand. DNA polymerase reads the sequence of bases on the template 3' to 5' and synthesize the complementary strand 5' to 3'. The following proteins and chemicals are required for DNA replication. First, DNA polymerase. Second, template. Third, primer. In vivo, primates synthesize the RNA primer. 4. Deoxynucleotide. Number 1 to 4 are required for DNA synthesis both in vivo and in vitro. Number 5 to 9 are required only for DNA replication in vivo. We will go over the list in detail later. Please check out this video for animation. First, let's look at primer. DNA polymerase cannot initiate new strand synthesis by itself. It requires a primer. When the primer is present, it is able to make a new strand. The question is, where can we get this primer? Answer lies in RNA. RNA synthesis can start without a primer, as we will see in the next chapter. A small piece of RNA generated by DNA primase serves perfectly as a primer. This can be better represented by this example. Here is a template. Using this template, DNA primase generates a piece of RNA that serves as a primer. With this primer, DNA synthesis can proceed. But this gives us a new task. We should remove this primer once DNA synthesis is complete, because this contains a piece of RNA. Removal of primer is accomplished by one of the DNA polymerases, which has 5' to 3' exonuclease activity. One by one, polymerase with this activity can remove the primer. We have a second task. What will happen if DNA polymerase make a mistake? For example, here is a mistake. To correct the mistake, we will need 3' to 5' exonuclease activity or proofreading activity. With this activity, this mistake can be corrected. Proofreading activity can also be represented in this slide. Bacteria have 3 DNA polymerases: DNA polymerase 1, 2, and 3. Not surprisingly, all have 5' to 3' polymerase activity. They all have 3' to 5' proofreading activity. But only DNA polymerase 1 has the 5' to 3' exonuclease activity. It means only DNA polymerase 1 can remove the primer. Initiator proteins are the key components of DNA replication machinery that determine where initiation will occur. Here is replication origin. In the first step, initiator protein bind replication origin. In the second step, helicase bind the initiator protein. In step 3, helicase is loaded onto DNA. In step 4, helicase opens the DNA double helix and binds primase. Step 5, synthesis of primer and synthesis of first DNA chain begin. In step 6, DNA synthesis in both directions start. Here is a replication fork. At the replication fork, one strand in red is being synthesized in the same direction as the advancing fork. This strand is called leading strand. But another strand is being synthesized in the direction away from the advancing fork, and this strand is called lagging strand. As the fork advances, this will leave a region that is not replicated in the lagging strand. This is a challenge. How can we solve this challenge? The answer is Okazaki fragment. Lagging strand is being synthesized discontinuously, with a small fragment being synthesized near the replication fork. These small fragments are called Okazaki fragments. Eventually, these fragments are joined together by DNA ligase. This is a full description of Okazaki fragments. DNA helicase separate the two DNA strands by breaking the hydrogen bonds between them. This will generate a positive supercoil ahead of each replication fork. It will make the advancing fork difficult to move ahead. To solve this problem, these two proteins help to keep DNA strands apart. First protein is DNA gyrase. This protein will travel ahead of replication fork to relieve the supercoil. Second protein is single-strand binding protein that bind the separate DNA strands. To keep the separate DNA strands apart. Here, this represents DNA gyrase. It is a topoisomerase II, which break and reseal DNA to introduce negative supercoil ahead of the replication fork. Here is the integration with pharmacology. Fluoroquinolones antibiotics block DNA gyrase in many gram-negative bacteria. This group of drugs include ciprofloxacin and levofloxacin. Put all players around the replication fork on one picture. This is an important picture. It requires students to understand where and what each player functions around the replication fork. One last question regarding DNA replication in bacteria is how we finish Okazaki fragment. Here the cartoon shows Okazaki fragments are being synthesized by DNA polymerase 3 or PO3. Each Okazaki fragment contains an RNA primer. DNA polymerase 3 elongates the DNA till it hits the next RNA primer from the previous Okazaki fragment. Then this enzyme goes off. DNA polymerase 1 takes over. DNA polymerase 1 initiates at the end of Okazaki fragment and further elongates the DNA chains while simultaneously remove the primer from the previous Okazaki fragment with its 5' to 3' exonuclease activity. Now, the two fragments are next to each other with a small gap. And this gap is sealed off by DNA ligase. These are Okazaki fragments with a small gap in between them. This gap is sealed off by DNA ligase. This step consumes 1 ATP. Now let's move to part 2. DNA replication in eukaryotes is very similar to DNA replication in prokaryotes, with only minor difference. First difference is on DNA polymerase. Instead of polymerase 1 to 3 in eukaryotes, DNA polymerase are alpha, beta, gamma, delta, and epsilon. Alpha has primase activity, beta and epsilon for DNA repair. Gamma replicates mitochondrial DNA, and delta is the core DNA polymerase. To assist you study this polymerase, here is a mnemonic. Alpha prime, beta repair, delta do the work. Represent alpha function as a primase. Beta and epsilon are used for repair, and delta is the main DNA polymerase. In the replication fork of eukaryotic cells, polymerase alpha synthesizes primer, delta synthesizes DNA. As you notice here, there's a protein that we did not see in prokaryotes, and this is a clamping protein. Clamping proteins holds polymerase to the template for rapid and continuous DNA synthesis in both the leading and lagging strand. Here, it shows you clamping proteins in the leading strand. In the lagging strand, every time when DNA polymerase delta stalls, when it gets close to the next Okazaki fragment, clamping protein dissociates and is reassembled with DNA polymerase at the next RNA primer. Eukaryotic chromosomes is linear and much longer than bacterial chromosome. For example, if human cells follow exactly the prokaryotic cells, replicating one human chromosome would require 35 days. This is the second challenge. The solution to this challenge, DNA replication initiates at many different replication origins simultaneously. DNA polymerase can synthesize DNA only in the 5' to 3' direction and cannot start DNA synthesis without a primer. But at the end of a linear chromosome, such as this, there's no place for primer. If this problem is not solved, the linear chromosome would become progressively shorter and shorter with each round of DNA replication. So this is challenge 3. The cells solve the problem by adding DNA sequence to the end of chromosome. These are small repeated sequences around 1,000 nucleotides. The reaction is catalyzed by telomerase. This enzyme contains protein and RNA. RNA is used as a template to synthesize a piece of DNA. Telomerase functions as a reverse transcriptase, an RNA-dependent DNA polymerase. This is how it works. In step 1, telomerase binds the end of chromosome. In step 2, it synthesizes 6 nucleotide short DNA sequence using the RNA as the template. In step 3, it shifts one position and make another 6 nucleotide DNA repeat. This cycle occurs many times. This greatly lengthens the end of chromosome. Now, cells can start to make a primer, and this is the primer. DNA synthesis can now proceed far away from the original end. This is integration with pharmacology. I have shown you earlier, quinolones are antibiotics by blocking DNA gyrase. Daunorubicin and doxorubicin are anti-cancer drugs by binding DNA and by blocking topoisomerase II. Etoposide and taniposide are anti-cancer drugs by blocking topoisomerase II. Azithromycin is an anti-HIV drug by blocking HIV reverse transcriptase, an RNA-dependent DNA polymerase, reminiscent of telomerase. Here is a azithromycin. How can it block the RNA-dependent DNA polymerase activity? It is a thymidine analog. Which element missing compared with thymidine make it a reverse transcriptase inhibitor? The answer is 3' hydroxyl group. Now let's move to part 3. 500,000 to 1 million individual molecular lesions occurs per cell per day. Unrepaired lesions in critical genes can impede cell function and increase the chance of tumor formation. DNA damage can be broadly divided into two classes: endogenous versus exogenous. Endogenous damage arises from internal metabolic processes. Exogenous damage is caused by environmental factors such as UV light, radiation, and chemicals. Here are common DNA lesions seen in patients: strands breaks, missing base, altered base, dimer formation, interstrand cross-links. For this lecture, we will focus on these three DNA lesions. Free radicals cause DNA strand break. In this example, both phosphodiester bond and the ribose are broken by free radicals. Around 10,000 purines are lost spontaneously per cell per day. For example, depurination releases guanine and adenine from the DNA. Missing base leads to deletion of nucleotides in daughter cells. For example, missing base will lead to deletion of a nucleotide in one of the daughter cells. Base can be modified by alkylation, typically methylation, oxidation, and deamination. Base substitution also occurs naturally during DNA replication. Positions affected are typically found at 1, 2, 3, 6, 7, 8 of the purine ring. Here is an example of oxidation. It occurs at position 8 of the purine ring, leading to 8-oxyguanine. This modification can cause point mutation. Chemicals can cause deamination. For example, cytosine is converted to uracil after deamination. Uracil can be paired with A, leading to point mutation. Very similarly, adenine can be converted to hypoxanthine after deamination. Hypoxanthine can be paired with cytosine, also leading to point mutation. Fortunately, deaminated cytosine can be repaired because uracil is not normally present in DNA. Nevertheless, if it is not repaired in time, mutation can be passed on to daughter DNAs. Cytosine is often methylated at position 5. If methylated cytosine is deaminated, it will be converted to thymine. This cannot be repaired because thymine is normally found in DNA. As a result, the mutation will be passed on to daughter cells. Two adjacent thymine bases can be joined together by covalent bond, leading to thymine dimer. Thymine dimer is induced by UV light. If DNA damage is not repaired in time, it causes senescence, apoptosis, or cancer. DNA damage can be repaired at 3 levels. At the first level, damage is repaired by proofreading by the DNA polymerases. At the second level, damage is repaired by mismatch repair system. At the third level, damage is repaired by excision repair system. Here we will focus on mismatch repair and excision repair. Mismatch repair systems fix the error made during replication, but not caught by proofreading. Mismatch repair system must be able to distinguish newly synthesized DNA strand from parental DNA strand based on methylation. Newly synthesized DNA strands are not methylated, but parental DNA strands are methylated. Several key proteins are involved in mismatch repair. For example, MLH1 and MSH2 are the key proteins that detect mismatch. We will see them again in cancer genetics. Parental DNA strands are methylated on certain adenine bases. Newly synthesized DNA strands are not. Mutations on the newly synthesized strands are identified by scanning for mismatch. After found, these mutations are repaired by excision repair mechanism. After repair, the newly replicated strand is methylated. Defects in mismatch repair cause hereditary non-polyposis colon cancer or Lynch syndrome. After cytosine is deaminated, it is converted to uracil. Uracil is not normally present in DNA. This base will be removed by uracil DNA glycosylase. Then AP endonuclease cleave an AP site to produce 3' hydroxyl group and 5' deoxyribophosphate. The gap is filled and the 5' deoxyribophosphate removed by DNA polymerase beta. Finally, adjacent two nucleotides are joined by DNA ligase. After the system detects a thymine dimer, nuclear excision repair mechanism kicks in. First, endonuclease cuts DNA damage on the 3' sides. An exonuclease cuts on the 5' sides. This leads to removal of around 30 nucleotides. Second step, DNA polymerases delta and epsilon fill the gap. Finally, two adjacent nucleotides are joined by DNA ligase. Here is clinical correlation. The first disease is Xeroderma pigmentosa. It is an autosomal recessive disease. This is a high-yield disease in NBME. Key symptoms: severe sunburn when exposed to only small amount of sunlight, many freckles, rough surface growth on the skin, and skin cancer. This is caused by mutation in one of the 9 genes involved in nucleoside excision repair. The second disease is Bloom syndrome. It features short stature and rash on the face. It is caused by mutation in Bloom protein, which is a DNA helicase involved in DNA replication. The third disease is Werner syndrome. It features growth retardation, short stature, premature graying of hair, alopecia, wrinkling, prematurely aged faces. It is caused by mutation in Werner protein, which is another helicase likely involved in DNA repair.